Hello, everyone. Welcome back to Creation Myths. I have a very exciting show tonight. Uh, but before we get into anything, I just need to make sure that YouTube is working, and it looks like it is. I will see in just a second. It looks good. It looks and sounds good. Chat, you know the deal. If there's any problems with the audio or video at any point, just let me know and I'll try to fix it. But right now, other than the fact that I look a little bit yellow, the lighting in here is a little bit warm. Other than that, it looks and sounds good. So let's get to it, everybody. Let me tell you what we are doing here tonight. I am thrilled because I have joining me Dr. Jay Wile. Jay did a show recently-ish. It was within the last few months on Standing for Truth with Donnie. And one of the topics they touched on was junk DNA, and Donnie spun that off into its own like half-hour video, which I watched because some of you may know I've done stuff with junk DNA in the past. So I reached out to Jay to see if he wanted to have a further conversation on junk DNA, and he very graciously said, sure, let's set it up, and got me on the schedule, and here we are. So Dr. Jay Wile has a Ph.D. in nuclear chemistry from the University of Rochester. He's worked as an assistant professor at Indiana University and Ball State University. He's a young Earth creationist. And I'm going to turn it over to Jay. Jay, if there's anything else you want to add, and then take like a few minutes to talk about your position on junk DNA, and then we'll go from there. And before you do that, just thanks for being here, everybody. Appreciate seeing all the people already here. Jay, take it away for a few.
Well, thanks for having me. You have a very pleasant personality, and I assume this, like you were on Standing for Truth, you're going to be a pleasant guy. And I always like to have pleasant conversations with people I disagree with. So looking forward to a good evening. I really don't have anything to add to that. Maybe that I'm a textbook author. I left my academic position back in the 90s to start writing textbooks. And one thing you need to do to write textbooks is to read a lot wider than your field of interest. And so part of my reason I left academia, I took a consulting position, which hopefully people know means less work and more money. And that allowed me to spend more time reading, to write textbooks and so forth. And that's where junk DNA first hit my screen. It was probably late 90s or mid 90s anyway. I hadn't broadened my reading very much, but I was reading Henry Morris's Modern Creation trilogy. And he was talking about pseudogenes. Now, I don't think he really understood everything, but he did say that, evolutionists say pseudogenes don't have any function because they can't be translated into protein. And he says, we're going to find function for these pseudogenes. And that kind of perked me up because as a scientist, what makes a scientific theory scientific is not that it explains things. Bedtime stories explain things. What makes a theory scientific is it predicts things, and those predictions can then be confirmed or denied. And so here was a prediction. Evolution said no function. Creation said function. So it was like, well, I'll kind of broaden my look at the scientific literature and see if anything happens. And nothing happened for a few years. And I do think I missed the first one, which was on like a pond snail or something. But eventually I found a reference that talked about this particular pseudogene, that its transcript regulated the transcript of the working version of the gene. So it was a regulator. And I thought, well, notch that up for creationism, because in the end, it was a prediction that was confirmed. And so, of course, I wanted to start looking for more. And so as time went on, I learned more and more of these studies that showed supposedly junk DNA functional. And of course, this all sort of culminates in 2012 with the ENCODE study. I don't think the ENCODE study is necessary, and I'll say why in a minute. But I think the ENCODE study is essentially correct. I think their definition of function is good. You will disagree with that. And I do think it's a good way of showing how much of the genome is actually used. And I do think that well over 80% of the genome is functional. Now, once again, I am not a biologist. You've forgotten more biology than I know. So I'm not trying to educate you. I'm simply giving you my version of this, right? So anyway, and of course, there were a lot of folks who use evolution as their creation myth, who don't like that. And of course, probably the most popular person or most well-known person is Dan Grauer. And he said, quote, if evolution or if ENCODE is correct, then evolution is false, or something like that. shouldn't have said quote because that's a paraphrase. And so he writes a 2017 paper that says, based on the fact that the more functional genome you have, the more selection there has to be, the upper limit for functionality is 25%. Now, interestingly enough, 2 years later, he has to write an addendum to the paper. He has to write an error because some folks caught an error. He was using the haploid version of the genome. They said, look, you got to use the diploid version. So he had to put a factor of 2 in there. That actually lowered his functionality estimate. Initially said maximum 25%. His new addendum says maximum 15%. So that error actually caused his conclusion to be more tight. And I don't think that number is necessarily the right number, but I do think broadly his conclusion is correct. That in the end, a highly functional genome is a real problem for evolution. But let's just get rid of the encode for a minute, because we don't need to worry about encode necessarily. Encode gives sort of a broad view, right? We can get rid of encode and just look at the trend of the data. The trend of the data is more and more function is being found. That's more and more in line with the creationist view, less and less in line with the evolutionist view. And this is why Maria Stitz and her colleagues back in 2021 said the days of junk DNA are over. She's not part of the encode. As far as I can tell, none of her team is part of encode. And they weren't working on something broad. They were working specifically on one type of junk DNA in one organism, a blood fluke. But their point was, we keep finding functionality for junk DNA. So the days of junk DNA are over. And I broadly agree with that. And I do think that is a difficulty for evolution, not only because of natural selection's difficulty keeping up with mutations, but also in general, it seems to me, at least, that evolution needs a genetic playground. It needs something that can mutate a lot to produce these wonderful genes. And so I would think there needs to be a pool of junk DNA available to evolution. So that's my basic position. Have at it.
Okay, thank you. And I wanna clarify a couple things just to make sure I have your position locked down, so I
Sure.
make sure I don't mischaracterize you. So you said you think that the genome, in terms of the percent functionality, well over 80% was what you said.
Yeah, probably 90 or more.
90 or more. Yeah. A lot. So,
yeah,
right. A lot. Like 80, 90, somewhere high, above 80, above 90, somewhere in that range.
Probably
way
above
up there.
90.
Okay. And if you had to assign like a percentage to that, are you 100% confident, 80% confident? How are you kind of feeling about that?
Well, as a scientist, you have to hold all complicated theories, especially complicated theories about new things, a bit tentatively. That's what a good scientist does, right? That's why I'm not the typical young Earth creationist. I don't say you have to be a young Earth creationist. I hold these views like a scientist should, tentatively, right? So I would say I'm more confident than not, but I'm not sure if I had to put a percentage on it, maybe 75.
75%?
That's hard to do. I would just say more in general, I'm certainly open to being corrected on this.
Okay. I do want to let me to contextualize this for everybody. In that show you did with Donnie with SFT, I just want to play a couple clips just to kind of contextualize your level of confidence in this and let you kind of just make sure we're on the same page. So let me. There's a very short. This is I think it's like 40 seconds or something. Let me add this. Chat, if you can't hear this, let me know. You should be able to hear it fine. Let me just do this here.
All right. But of course, now we know that's completely false. We know that the vast majority of the human genome is functional. So from the early 70s up until, shortly after 2012, but definitely by 2021, junk DNA was considered a scientific fact. And it was not only evidence for evolution, but it was the source for genetic evolutionary innovation. And we now know that's just false. The days of junk DNA are over. It's a recent example of the scientific consensus being dramatically wrong. I mean, not just a little bit wrong, dramatically wrong. It was the scientific consensus since the 70s that 98% of the human genome is now is useless. We now know that's absolutely false. And once again, the title's wrong. We know that. We know it's not 90% non-functional.
The
Well, I was
title-
laughing because that spelling was dramatically wrong.
The title in question, for anyone wondering, in that last little bit there was Larry Moran's book, 90% of Your Genome is Junk, was the title you're referring to. Okay, so you said like more confident than not. I will say you sounded pretty darn confident in that show with Donnie.
Well, I think, when you're talking about what's my scientific view, I'm going to give my scientific view fairly confidently, right? But like I said, all of my scientific views are held tentatively.
Okay. So let me, I told you I was going to do this. So let me to be fair, I put you on the spot, said, what's your, number for functionality? And it was over 80%, maybe over 90%, something like that. So it's only fair if I provide mine. And looking at I'll just say up front, I think that 80 to 90 or more number is biochemically and genetically basically indefensible. And I'm sure we'll talk about why. I think we have really solid evidence for functionality in 8%, maybe 10% of the genome. And I think we have reasonable inferential evidence to get you to 12, maybe if you stretch it, you can get to 15, is what I think the data are pointing to right now. That's where I'm coming from. So we've got a big gap here. We've got 80% or more versus like 10 to 15%. That's where we're at here, folks. So before we go any further, and again, Jay, I said I was going to do this in the green room before we started. Folks watching this either currently or in the future, there are a bunch of papers on this topic that are linked in the description. So I'm going to pull up just screenshots of the cover page showing the title and authors for each of those papers. So if anybody wants to see what they are, there they are. You can look them up. The links are down below. But I want to make sure everyone kind of here, us and the audience, is on the same page with this. So let me pull this up here. So the ones that I have linked down below, I have the famous 2012 ENCODE paper, an integrated encyclopedia of DNA elements in the human genome. This is like the big 2012 paper that made the giant splash, where the headline that everyone got was that 80%, we've been able to assign a biochemical function for 80% of the 80 whatever percent of the genome. And most of that was transcription, which was 80.4%. Then we have this was a really interesting one on purifying selection. This was Ward and Kellis 2012. Jay, have you seen this one? This is a good one. Yeah, we've got Ward and Kellis 2012. We've got Grauer's earlier paper, the 2013 paper.
That's
right.
a clever one.
And relevant to this is actually the phrase, the statement, if ENCODE is right, then evolution is wrong, which is from the SMBE conference in Chicago in 2013. And here's a fun little tidbit. I was at that talk. I was at the conference as a graduate student. I was at that talk. It was fantastic. And every creationist who has ever talked about that has gotten it wrong, because that's not what he meant when he said that. What he was describing was that if ENCODE is right about the amount of the human genome that is functional, then there's some basic aspects of population genetics that we need to rethink in terms of the relationship between drift and selection. That's what the phrase, if ENCODE is right, evolution is wrong, meant. It doesn't mean evolutionary theory is wrong.
Well, no one expects that. That's what it means. I mean, at least no one who knows scientists expects that's what it means.
No one expects our basic understanding of population genetics to be wrong. Yeah, that would be quite surprising. So then we have this one also. I think this is 2013. Yeah, this is the case for junk DNA, Palazzo and Gregory. This. Have you read this one, Jay?
I don't think so.
This one goes through basically several levels of the case for junk DNA. I think very persuasively. So that's a good one. Then we've got the 2014 ENCODE paper, Kellis et al. This one is, I think, reasonably well known. You probably have read this one.
Yeah, sure.
Yeah. This one is notable because it's an ENCODE paper and it's a significant walkback of the 2012 paper. Nobody ever cites the 2014. They always quote the 2012. They don't cite the walkback.
It's not really a walk back.
It was extremely a walk back. It was because they described very specifically the conditions of if we see XYZ, we think that DNA is likely non-functional. And then in the figures, those qualifications apply to like 2/3 of the human genome. So they also said, we're not going to say a number in this paper. It was a walk back.
No, it wasn't.
And then we've got Rands et al. 2014 looking at functional constraint in the human genome, about 8%. This is a really good one. Lynch and Marinov, bioenergetic cost of a gene. So this one, you actually mentioned the energy argument in your chat with Donnie, and this is relevant to that in terms of like if you have transcription or whatever, is that necessarily functional? Have you read this one?
Yes.
Yeah, this one's good. And then we have the famous Grauer 2017, which has now been corrected because the math was wrong. And then the Galeota-Sprung 2020, which is the follow-up to the correction. And the notable
Not really.
thing,
It's completely different.
it's the same people who found the error. And so
Right.
they're taking Grauer and they're expanding it, but doing the math in a way that they think is correct. And I've actually had a conversation with Grauer about this because he was a reviewer on this paper. And so he said, I reviewed their paper. They were right. I was wrong. And he doesn't even stand by the 2017 paper.
Well, he has
anymore.
never made that statement publicly in any way. I mean, I
He-
follow his blog. His blog is fun to read, a little juvenile at times, but it's fun to read. He's never said that in his blog. I've never seen any published indication. And even in his correction, he didn't say, hey, I'm wrong. In fact, he said, I'm even more right.
Well, he did actually, the table in the correction does actually have the numbers where it's like the number of kids you would have to maintain
Right.
the functionality. Those numbers are a lot lower than they were in the original table.
No, his number, yeah, the functionality is a lot lower. His original table makes the functionality even lower than his corrected paper makes the functionality even lower than his original paper.
No, it's the functionality doesn't change. The functionality is across the top. In the table,
Right.
it's the number of offspring.
Right,
and
exactly,
it's the
but-
number of kids to maintain a certain level goes
Exactly.
down once you consider diploids. It gets easier to maintain higher function
No,
when you
it's
have diploids.
exactly the opposite.
No, the tables are different. Anyway, this paper supersedes that, and he gave me permission to share the email exchange that we had, so I have a video on this where I showed he agrees with this paper, and in this paper they say very clearly, if you do the math, what these authors say is correctly, which Grauer endorses. They say, contingent upon the strength of selection, there is no upper limit to the amount of functional DNA.
Well, what they say is for their assumed selection coefficient, actually 0.5 or more is about the most you can have. That's what their table says. And in fact, their unpeer reviewed paper. No, that's what their table says. Their table in that paper says that at a selection coefficient of 0.01, after 0.5, you've already got a 4.88 W bar over or W max, which is already a very deteriorated genome. And in fact, in their unpeer reviewed paper that's still on the archive, it actually says in the conclusion, the functionality can be 0.5 or perhaps a bit higher, but no more.
And in
so
the published
they're still
paper,
putting a
so
link-
that's the unpublished paper, though, you
Right,
said. It
sure,
could be
yeah,
0.5 or a bit more. In the published one, the specific line is that there is we don't identify an upper limit.
Right, we can't identify an upper limit.
Right.
That's,
Yeah. And then the last thing on here, I don't have this link down below because it's a book, not a paper, but What's in Your Genome? 90% of Your Genome is Junk by Larry Moran. Have you read this one? This is a good... No, you should read this book. It's very good. And if you have strong opinions on junk DNA, you should read this book.
Well, I read Sandwalk.
Well, you should read the long version. It's good. Okay, so those are resources that I think everybody should have. And like I said, everything but the book is linked down below. So now we've established kind of background here. We've got a bunch of sources. So I want to go through with you just kind of what all this function is in the genome, right? So I want to just hand it back to you for a few minutes. If you just want to tell us what the genome is doing. What is happening in this 80 or 90% of the genome that's functional?
Well, I mean, there are lots of different components. So there are regulators, pseudogenes, of course, do regulation. Others, like, for example, was called genetic stuttering. I can't remember the name right off the top of my head. Mini satellite tandem repeat sequences. those were thought to be genetic stuttering. We now know those are regulatory. The more stutters that exist, the slower the expression, the less, and enough stutters can actually turn off a gene. So we know, for example, there's a lot of regulatory elements, but we also know there's a lot of structural elements that are absolutely necessary. The DNA has to be ordered very specifically while it's in interphase. It has to be ordered differently when replication is starting. So in the end, there are structural elements that do that. And so some of the things that are transcribed at extremely low levels are used specifically to make sure the centromere is in the right place, make sure the DNA is connected to the kinetochore, and so forth. So you've got structural elements that are really important as well. I also think that as time goes on, you're going to find that, well, like for example, what is the one that Stitz they were talking about satellite-like W elements, right? And they're talking about how there are mobile genetic factors that actually help the organism adapt to stress. And we know that's true of certain other transposons, that they actually are sort of pre-programmed mutations, if you will, not exactly that, but they're pre-programmed reorganization of the genome. I think we'll find that the genome is a lot more flexible than we currently think it is. And I think a lot of this functional DNA is that. I also think that a lot of the transcription that ENCODE is seeing is actually simply protecting against mutations, because we know there's transcription coupling repaired, transcription coupled repair, and a lot of these structural elements, they need to be transcribed just to keep from being mutated in a very serious way. And so I think a lot of this transcription that's going on in the ENCODE project is actually simply protecting the structural elements properly.
Okay, so we got a few there. Pseudogenes regulating the kind of similar active gene.
As RNA
repeats that are regulating transcription rate. Structural elements, and that could be telomeres, centromeres. You've got spacer regions where, like, between a promoter and an enhancer, it's got to be a certain distance. The sequence doesn't matter, but it's got to be a certain distance. Also, you could throw in there like histone attachment and methylation targets, I guess.
And telomere caps.
Telomeres, of course, have to be part of it. It's a small percentage, but that's you need those. You super duper need those. Yeah. Mobile genetic elements, so like transposons and retrotransposons are the big one, because that's like 44% of the genome is retrotransposons. They're doing like you mentioned stress response, but other people have mentioned. I like how I'm making your case for you here. You've got they could use the promoters. They could do interfere with the mRNA either up or down depending on the circumstances for their transcript products. You didn't mention ERVs, endogenous retroviruses.
No, I didn't.
But people have proposed like antiviral functions there, where it's like the real virus and then the fake transcript. It interferes with it. Yeah, I think that's a pretty good look. Plus, of course, like protein coding DNA, promoters, enhancers, silencers, ribosomal, RNA genes, ribosomal RNA, transfer RNA. We know those, small interfering RNA, right? Everyone knows those. So those are all the functions that we can.
As well.
I'm sorry.
Mini-sats.
Yeah, right. All the right signs and lines, like the little repeat elements that are all over the place. And I want to be clear about this. In my 8 to 10 to 12% number, we've clearly identified function in every one of those classes that we just named: pseudogenes, ERVs, retrotransposons, and within that, signs and lines and this ALU elements. There are members of each of those classes where we have conclusively said, here's a specific member of that, here's an ERV that does this specific job. Here's a pseudogene that does this specific job. Like, those are things we have identified. The problem is that most of each of those classes are not doing any of those jobs, and we can be very, very sure of that.
See, that's where I disagree.
So let's talk about this. So we just went through all these functions, right? Protein coding, regulatory, structural, all this stuff. The key thing that allows us to identify all of these elements in the genome and any functions that they do is their sequence, right?
Right.
Right. It's like if you have a gene,
but not all of them.
Well,
not lots of non lots of functional DNA is not sequence limited.
Okay. So let's go through what is and isn't that. So like protein coding genes, pseudogenes, regulatory elements,
some regulatory elements. Not all regulatory elements are sequence specific.
Right. Yeah, no, I'm going for sequence specific things, right?
Okay. All right.
Protein coding genes, RNA genes, pseudogenes that are doing some regulatory thing, any ERVs that are doing antiviral stuff, telomeres, centromeres, anything binding, right? Like a histone binding site, methylation targets.
Now, except there are these RNAs that they bind not because of a lock and key mechanism, but because of these fuzzy motifs. So if there's if we've got this RNA that's just got this guanine-rich region, that guanine-rich region can move around.
So you're talking about is that a transposon, a pseudogene? What is that?
Well, let's see.
It's like, what does that do?
I'm not sure I remember the. Here we go. It's Kamer clustered long non-coding RNAs.
Okay, and what's the function for that region?
So they act as architectural scaffolds to recruit proteins, but they don't use a lock and key mechanism.
Okay, and what percent of the genome are those?
Well, I guess it depends on how you look at it. When I made my list, let me tell you what my list is. I think I grouped them with something else, though, is the problem. 15 to 20% are genomic scaffolding and nuclear architecture.
Mm-hmm. And those are, so let me keep going. Okay, so we got 15 to 20% you're saying are these, and those are, what class of DNA are those, though? Because you're telling me the function, but what class of DNA? Because, right, we've got like exons are 1.5%, right? Retrotransposons are 44%, ERVs are 8%. DNA transposons are a couple percent, pseudogenes are like 5%. Like which of those things are those?
Well, they're long non-coded. They produce long non-coding RNAs.
Right,
so,
but lots of those do.
Yeah, but I really don't know. I don't know specifically how you'd say them.
But there's something transcribed.
Yes,
they're transcribed. Okay.
They make make a long non-coding RNA, but it's not sequence specific because it's not using a lock and key mechanism.
Right. But it does require a certain, it sounded like it's guanine rich, so it needs to have a certain percentage of guanine.
But only sections of it need to be guanine rich. So the idea is the sections that aren't guanine rich.
So the spacers, so that falls into your spacer. Yeah.
Or those guanine rich sections can move within the link RNA and still do the function.
Okay.
So the sequence is not very specific at all.
So follow up on that, is it subject to purifying selection? Like if it mutated, is it still going to do its job?
Well, as long as, like, for example, if the guanine-rich section simply moves, then yes. Or if the section in between the guanine-rich sections mutate, then yes. Right? The only mutations that is going to cause it not to work is when the mutations hit the guanine-rich.
Hit the guanine. So
and I'm sure you know more about this than I do.
I just want to follow up there. So the guanine-rich regions, you need the guanine, so that's going to be subject to purifying selection. Sure. The other parts is in kind of that what I call, I don't know if it's like even the right word for it, but I call it spacer DNA, where it basically just means you've got this thing and you've got this thing, and they need to be separated by a certain distance, right? Mm-hmm. And what is in there doesn't matter.
Yeah,
but it's got to be there. Yeah. Or another example of this is, you have like a promoter and an enhancer, and it has to be, 1,500 nucleotides. If it's 1,000 or if it's 2,000, it doesn't work. But if it's the right range, you need the right range. Okay. So here's the key with all these functions. And so you've got, like we said, protein coding genes, RNA genes, regulatory pseudogenes, any promoters, enhancers, silencers, any of the regulatory regions, structural stuff, telomeres, centromeres, any of that stuff, methylation sites, histone binding, anything like that. All that stuff needs to be a specific sequence in order to work, right? Like those are things you can't mess with.
Well, except that we used to think that pseudogenes were unfunctional because they had the wrong sequence. Turns out they have the right sequence for their job. So when you say it needs to be sequence specific, the question is, what sequence?
Well, the sequence it has to do its job.
No, well, but pseudogenes are a direct conflict with that. Pseudogenes don't have the right sequence, and yet they still work.
Well, you say they don't have the right sequence, by which I assume you mean they don't have the same sequence as the functional gene.
Well,
right?
that was the original argument, right?
Well, right, but how are we defining right here then? Because if it's functional with a different sequence, then what does right mean in that context is what I'm trying to get at here. Because you're saying they have the wrong sequence. That doesn't mean they're not subject to selection to maintain the sequence they have if that sequence is doing an important job, is what I'm saying.
Right, but the idea is the sequence can be different in some way
Right.
we're not expecting, and
Right.
still be functional.
Right.
I mean, pseudogenes showed us that.
Well, let me ask the same question a different way then. Could any sequence do the job that a regulatory pseudogene does?
Any sequence, not like a
Yeah.
bunch of T's, no.
No, right. So there has to be some level. So the point is there has to be some level of purifying selection operating on that sequence to maintain that function. If it mutates too much, it's not going to match the target mRNA.
Well, but once again, not all regulators use a lock and key mechanism. So the idea is, what's your limit on?
Well, do
what's
you
the right
have,
sequence?
so for pseudogenes, do any of them operate through a kind of more mushy affinity mechanism rather than a sequence homology
I don't know.
mechanism?
I don't know. I mean, it could be that a pseudogene has been recruited to be a mechanical spring, in which case it doesn't need to have a sequence at all. It just needs to wind and unwind properly.
Has that been
to release
directly
stress.
documented?
No, I don't think so. But my point
it
is,
hasn't.
that's something we're not even considering. And the fact is, before we didn't consider pseudogenes as being functional because we didn't consider a regulatory component. We've got to consider the things. I mean, this is a
Well,
fundamental.
hang on. I want to get at a question that actually you just referenced something that goes back to something you said earlier. Historically, what percentage of the genome did we think was functional?
Well, initially, at least the way I learned it in the biochemistry class I didn't like in college, it was about 2% was functional.
So you were taught wrong. That has never been correct. Because 2% was for a long time thought to be the protein coding regions. It's like 1.5-ish%, maybe a little lower. But that's the number historically you get for protein coding. But the problem with that is we identified non-protein coding regions with well-defined functions prior to the use of the term junk DNA. So like promoters and ribosomal
Except
RNA.
those were considered very small percentages.
Transfer. Well, it's quite a bit more than 2% once you put all that stuff in.
Nowadays, we recognize that.
We documented tRNAs before we even knew like how the genetic. I think it was like way back in the 50s. So we documented these things before the term junk DNA was actually in wide use, and certainly before it appeared in the literature, which wasn't until the early 70s. So it's never been the case that biologists were going around saying only 2% of the genome is functional.
The biologists that taught me were, and the biologists that write in the literature and write popular
books
do, because I've read it.
As we ought to know, the biologists that write the popular literature are often very, very mistaken, as ENCODE has taught us. So here's my point. We've got a couple of functions that can be independent of sequence. You've got certain types of associations that can be non-sequence specific affinities. You have spacer regions that are non-sequence specific. And those add up to a few like single digit percentages. I think the scaffolding argument is not good because there's I don't think those assays are, I think they have high sensitivity and low specificity is the way to say it. I think there's a lot of false positives in those scaffolding assays because biomolecules are by their nature sticky. And just looking at associations between pool of molecules A and pool of molecules B is not actually an informative approach to learning if there's functional significance to those interactions. You need to do other biochemical assays to solve that. And there have been some examples where there are like structural regulating the structure of DNA in the nucleus kind of stuff. But it's not 15 or 20% has been demonstrated to that level of confidence. The 15 to 20% number is highly speculative. If you,
I would disagree with that, and I think we know, for example, topologically associated domains are very important. G quadruplex complexes are very important, and those are all structural.
And those are very tiny fractions of the genome.
My number says a lot more than that.
The 15 to 20% number is significantly higher. than what like specific biochemical assays have demonstrated on that. But I don't want to get stuck on that, no pun intended, chat. Because I want to focus on all the other functions here, because that's just one of, like, the structural stuff is just one of all of these functions, and all of these functions, I think you would agree, require some level of sequence specificity, right? You can't have a protein coding gene that can be literally any sequence. It won't do its job. You can have some mutations, but at some point, that protein is not going to do its job. You can
Yeah,
have
but what does
promoter.
at some point mean? At some point, meaning 90% of its sequence has been degraded? I mean, in some of these cases, all you have to be is guanine rich, for example. That doesn't take much sequence
that's the
specificity.
fun part about evolutionary biology, because we can actually answer that question by looking at the level of constraint in the human genome, right? We can actually look at what parts of the genome are being acted upon by purifying selection to stay the same, and what parts of the genome are allowed to mutate without carrying a cost, right? What parts are just allowed to wander through sequence space with no cost, right? That's a question we can answer, and we know the answer to that question. A couple of the papers I showed earlier are examples of studies into that. One of them said 8.2%, with the range was like 7 point something to 9 point something. And then the other one was about 9%. And the way they did that was they said 5% conservation in mammals, but then looking at humans, they identified an additional 4% that has a lower substitution rate compared to other parts of the genome, indicating that natural selection is operating via purifying selection to keep that region the same.
Yeah, but
right?
these are all making relatively large assumptions about what it means to be sequence specific. You're saying, okay, if the sequence stays the same between rats and humans, then it's highly conserved, and that's not a good measure.
Not what it's saying, actually. These studies were not just looking at comparisons across the different groups. They were looking for signatures of purifying selection in the human genome.
Okay.
So you're looking at relative substitution rates across different regions of the human genome, which, chat, I know some of you probably know this, some of you might not. So a mutation rate is the rate at which mutations occur, right? So mutations are stochastic; they're just going to happen.
No, that's not true. We know that.
Mutations are stochastic. That's a different thing. It's not specifically related, but we've known that since the 1940. Luria-Delbruck study. Yeah. Like, we've known that mutations are stochastic since the Luria-Delbruck study in the 1940s, the fluctuation test with phage resistance in bacteria. And then the follow-up study, the Lederberg study, which used replicate plating to show that it was actually a stochastic distribution. So, like, mutations are stochastic. That's, like, not open for debate. We've known that.
It is. I mean,
years.
We know there are hotspots. Every gene has.
Yeah, there are hotspots, but you can't make specific mutations happen, right? Like a bacteria exposed to a bacteriophage can't evolve resistance due to that exposure.
I mean, Scott Minich's experiments indicate that's not the truth.
No, so you're referencing the Van Hofwegen paper. Looking at, that's the Scott Minich, the Lenski Sit Plus paper, right?
Yeah, right.
Yeah, where he was able to re-evolve it really quickly. Yeah. So the reason it went faster in that paper is twofold. And chat, sorry for the digression. We will get back to junk, but this is worth doing. So the reason that it evolved faster in the Van Hofwegen study is twofold. One, they used a stricter selective pressure. So in the original Lenski, it was glucose diet with citrate present. So you got a boost if you could use the citrate, so you could consider it like a screen for citrate. Sure. Whereas in the Van Hofwegen, I think Scott Minich was the senior author on that paper. Right. They made it basically so that if you can't use citrate, you're going to die. So that was like a filter for citrate metabolism.
Sure,
so,
and that makes sense. I mean,
right. So
that's why they got it go faster.
The other thing is they didn't use the original 1988 bacteria, right? So in the original Sit Plus, in the original Lenski experiment, they started in 1988, and it was around the 30 to 35,000 generation mark.
31,500.
That you saw that big increase in density in the one population. Now, what they found going back is you have earlier mutations that were required. And if you just do the duplication without the earlier mutations, those bacteria actually die. The duplication on its own was harmful. You needed the earlier mutations. And in the Van Hofwegen paper, they started not with the original, they started with one of the further along ones. So it had some of those priming mutations already. Those two factors together explain why it happened faster. It's not that the bacteria were deciding or responding purposefully. It's that the
No, it was that it was
conditions
an
of the
adaptive
experiment were different.
mutation. It was an adaptive mutation that was pre-programmed into the DNA.
No, there's no. Well, adaptive in that it was beneficial, but that does not, adaptive does not mean pre-programmed. Those are two different things. I mean, if it was pre-programmed, why didn't the other 11 Lenski lines do it?
Because the selection pressure wasn't high enough. You have to stress the cell enough to get this pre-programmed response.
They all had the same selective pressure. All 12 lines were the same environment.
In
the Lenski experiment.
In the Lenski experiment, right.
So
but once again, they did the glucose plus citrate.
Right, but if it was, no. The whole experiment was run glucose and citrate. Citrate was a pH buffer in that experiment. All 12 lines had the exact same selective pressure. Only one of them evolved Cit+.
Right.
Was it your understanding that only one of them had the citrate?
No. But what I'm saying is there wasn't enough stress, so you didn't get the pre-programmed response at a higher percentage.
What's the evidence that it's pre-programmed?
The fact that Linsky or that Minish could repeat it.
But I just told you that Minish started partway along
He had to get the rest of the way.
Sure.
And the
because
rest of the way
mutations are
was
stochastic, and if you have a giant bacterial population, I don't
No, because in the end,
hang on, please don't interrupt. Please don't interrupt while I'm mid-sentence there. You're using the existence of a beneficial mutation to say, therefore, it's pre-programmed. What you would need to do to demonstrate that mutation is pre-programmed is to show that the distribution is non-random.
And that's exactly what the Minish paper showed.
But they're using different. You can't compare the Minish paper to the Lenski study because they're using different conditions, is what I'm saying. The Minish paper,
I'm not comparing,
uses a much stricter filter for the trait, right?
Agreed.
Whereas the Lenski study, it's an option, but like you can live without it, which is right.
However,
what I'm saying is you need a mathematical distribution showing that mutation happens statistically more frequently than other non-adaptive mutations, and that's not in the Minish paper. It's not there.
That's what the Minish paper shows by the number of times he could replicate that and the few number of generations it took.
That's not because he doesn't have a control. See, the thing is, he's not controlling it appropriately. You're assuming that mutation is a response to the environment, right? That's the conclusion, right? That mutation is a response to the environment. So that's wrong. We know that's wrong. One, because Minish didn't actually show that. Like mathematically, there's statistics you need to do to show that. He didn't do that. The earlier studies on this exact question, right? The Luria-Delbruck test and then the Lederberg follow-up with the replicate plating, they actually show the math in both those papers. Here's what the distribution would be if it was a directed mutation. Here's what the distribution would be if it was stochastic mutations. And it's 1-to-1, it's the stochastic distribution, just straight up. Like, I want to emphasize to everybody the degree to which this is not an open question, like not even a little bit in bio. Mutations are stochastic. There are hot spots.
Yes.
There are hot spots. That is true. But you cannot pick a specific mutation. You just, like, that is a thing in bio. An organism cannot direct a specific mutation to happen. Like, just straight up, like, that is, the sky is oranges.
Not what the Minish paper shows.
That's not what the Minish paper shows. Anyway, let's continue. Okay. So we have the genome. The genome is 8 to 10% constrained, depending on the study you look at. It's about 8 to 10% constrained. Most of the functions that we've identified, that ENCODE has identified and follow-up studies have identified, require sequence specificity. Right? Most of those functions require sequence specificity. All of them will be subject to purifying selection. And actually, one of the papers, the one that was on the 8.2%, actually does include the spacer regions, because that was looking at purifying selection against deletion. So it wasn't even looking at the sequence. It was more inclusive than that. So the 8.2% was actually including regions that aren't sequence specific, but you need to have them. So that would include like a spacer region, a non-specific scaffold region. That's in there. That's caught in that study, right? So here's the thing. If we've got 8 to 10% of the genome, and I'm just going to say 10%, round up just to make it easy. if we've got 10% of the genome that is required to be there, right? There's purifying selection against deletions, right? If there's purifying selection against deletions, you need it, right? You can't get rid of it. That's fair, right?
Yeah, fair.
What function is the rest of the genome doing where there's not purifying selection against deletions?
Well, once again, it could be a lot of structural stuff. It could be a lot of non-sequence specific stuff. There's all sorts of functions going on in the genome that do not require a high degree of purifying selection.
But if there are functions that are like useful for the organism, right? And you delete the region that does that function, will that have a cost?
Well, yeah, I would think so, but you have to test that you have to test that properly. Like you do these knockout experiments. These knockout experiments
Right.
are not good tests for whether a region of the genome is required, because in the end, it's been shown that wild type mice do better than these knockout mice when they compete against each other.
Okay.
We know that. So in the end, it's very difficult to say, okay, I'm going to knock out this region. This is much like the old evolutionary idea that the appendix is useless.
So wait, but I think so.
Please don't interrupt me when I'm talking.
Yeah.
Right. So the old evolutionary idea that we can take the appendix out because we've taken it out of people and it's not done any harm to them. And so it actually became standard procedure to remove the appendix if it presented itself. And now we know that was false. We now know the appendix is extremely important. And so this is the same principle. Okay, I'm going to take out this part of the genome and it's not going to do anything. Well, that's just like the appendix. Yeah, you take it out and you don't test it properly. You're going to not see its importance.
That's what these papers did, though. That's what I'm saying, right? The papers are evaluating the genome, not based. They're just saying, is there a cost to losing this thing or not? Right. That's all these studies are saying. Right. It says there's a cost if you lose.
But they're testing that in a specific way.
Yeah, there's a way to test that. It's what you look for. Purifying selection. That's so you can look at the substitution rate, which is how you do that. So you look at different genome regions to identify whether deletions are tolerated and whether mutations are tolerated. Like you're asking for a test of how we can evaluate that. I'm telling you what the test is. Like that's what that is. So we got a little. Actually, this is a good place because a minute ago, hang on, we got a little sidetracked from the definitions here. So I was saying mutations just occur, right? And again, like they're stochastic, they just are. And then the mutations that achieve fixation, those mutations, that's called the substitution rate, right? So the mutations that become fixed in the population, those are substitutions, right? So what we can do is we can look at the relative substitution rate, both in terms of point mutations and things like deletions across the genome. And we can look at the regions where if you have either mutation, like point mutations or deletions, there's a cost associated with that versus regions where there's no cost associated with it. Like that's the test. That's how you do that kind of thing.
And how do we measure the cost?
By looking at the relative substitution rate. Because if you have a region that across all of humanity, for example, has a lot of variation, right? You've got a higher substitution rate because in the different lineages, you have different mutations. Yes. Good. So you've got a higher substitution rate. That means there's no cost to having those differences because they exist and everyone's doing fine with them. That's how that. Now you look at a different region, a highly constrained region, and you can say, oh, this region is remarkably consistent across all of humans. There are no deletions. There's no tandem repeats. There's no point mutate. Right. There's no variation in this particular region. That region is highly constrained. It's indicative of strong purifying selection, right? That's how we make that determination. You can look at relative mutation accumulation rates across different parts of the genome.
Yeah, but just saying that it has more substitution across, the human populations and so forth doesn't mean there's no cost to it.
I mean, that's literally what that does mean.
No, it doesn't. Well, I mean, I don't know how you define cost. All right, but in the end, we don't know that just because it changes widely throughout the human population, that there's not some low level lack of or low level cost to fitness.
But then it wouldn't be as at the frequencies we observe, right? That is actually the test here that I'm trying to get across, right? If there was a cost to fitness, it would necessarily exist at a lower frequency, right? That's the test. Because That's what fitness is. If there is a cost to fitness, its representation in the next generation goes down. Right? If it is a lower fitness than average, then its frequency will decrease over time. So you have regions of the genome that exhibit very low variation, indicating that mutations in that region are costly because they decline in frequency when they occur. You have other regions where you have a lot of variation, indicating that when mutations occur, they can persist. There's no problem. There's no cost.
Or you have other regions that have a lot of variation because that variation is necessary.
I'm not sure I understand the point. So
well, because you and I have a different view of the genome. You take evolution as your creation myth. As a result, there's no...
Okay, hang on. All right, no. Here's the thing. We're not going to do that. We're not going to do evolution as a creation myth. We're not going to play those games. Okay.
It's the name of your channel.
I know because that's what I do here. But when we're talking, we're not doing the name calling thing here. So the evolution is a creation myth. Evolution is faith-based, evolution, right? All this stuff. We're not doing that. I just want to make that crystal clear.
But you're doing that every day with your channel. You're saying I believe in a myth, right?
Yes, because
so you're calling me a name.
Well, I'm not calling you a name.
I can't. Well, you're claiming I'm calling you a name by saying that evolution is your creation myth.
Oh, right, and I'm saying that's not what we do on this channel.
Well, but you do it every day by saying that mine is a creation myth.
Yes. Yes, so, I'm not saying that our two things are equal here. I've got the science on my side. Young Earth creationism doesn't. That's what I do here.
I would disagree with that.
You're welcome to, but you're not going to be rude about it, is what I'm saying.
Well, but you're already being rude about it to me.
Have I been rude? Chat, have I been rude in this conversation? Chat, tell me, have I been rude in this conversation?
How have I been rude? All I'm doing is using the same name of your channel to talk to you.
Okay. So, I'm going to pull up a clip from the
most evolutionists aren't going away.
I'm going to pull up a clip with the thing from Donnie here that's relevant. So I just want to show this just to contextualize this for everybody here. So here we are.
Now, most evolutionists aren't going away from it because it's a faith more than it is a scientific theory.
Yeah, so this is the double talk that evolutionists love to give. And what he said was evolution is just this smorgasbord of different possible explanations. And whenever you come up with some observation, you choose that one and that one. They fit together to make this observation. Another observation, we choose that one and that one. We fit them together. In the end, we always can explain away anything we want because we keep making these stories that often contradict each other. I mean, you really have to do some intellectual gymnastics to be an evolutionist.
Okay. Had that just.
Well, and that's essentially what you say about creationists. So what's the difference here?
The difference is I'm using science to back up my position and I'm presenting it accurately. And you're saying things like adaptive mutations are purposeful or directed. And again, experiments from 80 years ago show that that's not the case. So, that's just what it is. That's just we have so much I've personally done, I will tell you, I have personally, as part of my research, have done what's called a mutation spectrum experiment, where you look at just all the mutations that happen in lineages. I was working on viruses. And I can just show you the distributions, the mutations are stochastic. That's just how it is. And it's not, that's not up for debate. So what I want to get back on track here, is this, because the question I asked is this: if you have a genome, right, where you've got 8 to 10% of that genome is constrained, where if you delete it, there's a cost associated with it. Now we're getting hung up on how you measure that cost. And the way you measure the cost is by looking at the substitution rates across different regions. This is just how you do it in evolutionary biology. You can look at a thing and you can directly observe its fitness cost from one generation to the next. And you do that over long periods of time. You can see that the things that have a fitness benefit are either maintained if they're old, or they increase in frequency if they're new. And the things that have a cost will decrease in frequency over time. So the regions that are highly constrained, and nothing I'm saying is controversial here. Everyone, creationists generally agree on how natural selection works. That's all I'm describing here, right? Is if you have mutations or deletions, point mutations or deletions that carry with them a cost, those things are going to be lower in frequency over time than mutations that don't carry a cost, right? We should be able to agree on that, yes?
I'm not sure I can.
You can't agree on, I mean,
No.
that's just how natural selection works.
Well, but I guess my question to you would be, if you're so confident that there's no controversy here in biology, why are there so many evolutionary biologists who disagree with you on this?
There aren't. There's no one disputes what I just said about how natural selection
on,
works.
on the junk DNA issue.
Oh well,
I mean,
we'll
there
get
are
back to
lots of
junk DNA in a
evolutionary
minute. We'll get to that in a minute.
Okay.
But just on the mutation thing and natural selection and like how you detect selection and constraint and stuff, like everything I just said is completely uncontroversial, and the only people who really disagree with it, and I didn't ask you about this beforehand, so maybe this is your position, are like the continuous environmental tracking people. With it's mostly Randy Gluza at ICR. Are you kind of more in that camp with the continuous environmental tracking stuff?
No, I just think
Okay.
that our view of the genome right now is still fairly naive, and so there are a lot of things that we say clearly show this section is not important that is wrong. And I think that the trend has shown that already.
Right. Oh, the trend has definitely shown that we can increase we've increased the amount of function that we've identified. But how much of the genome right now has like a clearly defined function where you can point to a region and say this region does this job?
Oh, probably not very much. I don't really know what the number is.
Yeah, it's like 8 to 10%.
Yeah.
Yeah.
But what we've always said that number is low, and it's always increased because we've always found new regions that have function.
Well, has it
And this
always
is why
increased?
Spitz
Because
et al. Said
what did
today's
ENCODE
junk DNA
report?
are-
What did ENCODE report in 2012?
What, that 80% of it was transcribed and therefore functional.
Right, but transcription doesn't indicate function. That's silly. Like,
Well,
that's
I mean, I disagree with that. I think their definition of function
go
is
for
really
it.
good.
Why
Well,
does
because
transcription
the cell
indicate
is sort
function?
of a paragon of efficiency. I mean, the F1 unit of ATPase is 100% thermodynamically efficient, not even any friction, as far as we can tell. So in the end, we've got this incredibly efficient cell, and you're telling me that it's spending all this energy transcribing?
That is exactly what I'm telling you. Yes,
See,
because that was shown in that 2015 Lynch and Marinov paper, the bioenergetic cost of a gene.
Oh, I read that paper.
Right,
Yeah,
and
it
so
doesn't
they
show.
showed that the cost of maintaining DNA and doing error correction, and replicating the DNA, and most transcription is below the drift threshold for the human population.
Sure, but still, nevertheless, we're always going to.
That's it then.
An efficient machine is going to save as much energy as
But
possible.
but that's
even
why.
if it's
But
small.
here's the thing: do you know when I say drift threshold? Do you know what that means?
Sure.
Yeah. Okay. So if you have a thing that happens, right, and it is below the cost to it is below the threshold that you can select, then you can't prevent it from happening, right? Natural selection will not weed out the individuals that do that thing in favor of the individuals who don't, right? That's what that means. So it doesn't sound like you're disputing the findings of that paper. Is that correct?
I'm not disputing the level of energy that they suggest, but what I'm disputing is that means the cell would go ahead and waste that energy.
Right, because it's below the drift barrier. Yeah.
Well, that's assuming that this thing isn't designed.
Well,
I think
no, it's looking.
that's
not
design
true.
machines are efficient.
That's not true though. It's not assuming that, because you can actually look at the selective pressures operating. You can actually measure the fitness costs of having this amount of energy expenditure versus this amount of energy expenditure. Mm-hmm. And given the population, the effective population size, you can actually calculate where that threshold is and show that that difference is below the threshold. So there's no assumption there that it's not designed. We're simply directly observing. We're directly observing the energetic cost and the threshold of a cost that you could select. It's directly measuring two things and showing that one is lower than the other. That's all it is. There's no assumption of non-design there.
Yeah, the assumption is that just because it's below what we consider the selection threshold, that means it's irrelevant. That's
what do you mean by? Well, hang on. Do you know, like, we calculate that though, and we can directly measure it. Like, we can validate that. You know that, right?
Well, we can validate it within the parameters of our model. Yes.
Well, but then you test your model against the real world. right? So you say, if we have a trait X, Y, and Z, we expect to see selection. If we have trait X, Y, we expect no selection, right? And you can test that. And you can go out and look and see in organism A, given the effective population size, we expect to see, X amount of transcription in the genome. In organism B, given their effective population size, we can predict a different level of compactness in the genome, right? Those are things we can and have done, right?
Right, but that still doesn't
Yeah.
tell me that the cell isn't going to save as much energy as it can.
Well,
whenever.
it literally does, though, because if you can't select below the threshold, then you can't save
Once
the
again-
energy. That's what we're trying to get. Like, you can show this experimentally. I'm not describing anything that's controversial.
Well, it's controversial to me. Let's say it that way.
That is a bespoke position, I will say, because this is like, for example, we can make very particular predictions about genomes. Like, you can look at the human genome, and with just knowledge of the cost of transcription and the effective population size, you can actually predict how much spurious transcription you would expect. And those predictions look a lot like the transcription documented in code, right down to the amount of each transcript per cell. Because you could actually, its low-level transcription is probably spurious, but high level is going to be above, the energetic expenditure will be above the threshold.
But once again, we experimentally know that's not the case, because we know there's burst transcription. There was a great 2008 paper that was looking at yeast, and it showed that out of 100 cells, the average number of transcripts was less than 1 per cell. But that was because at one time, one cell had most of the transcripts, the other cells didn't have any. At another time, that cell didn't have any, the other cells had more. So there is burst transcription. And so there's no way you can just simply say, well, this level of transcription in the ENCODE project is spurious, because in fact we've got lots of different experiments that show very low levels of transcription that are for very functional genetic elements.
So two things with that. One, in the ENCODE stuff, you can't do the thing where it's an average, but in like one of the cell types, it could have been higher, and then in all the rest, it would be low, and you get the low average, but it's still a lot somewhere else. So you can't do that because their data don't show that. I actually dug through the supplementals specifically on this and went through, literally, there's a supplemental on one of their papers where it has, and I'm not joking, every transcription level assay that they did in the supplemental, and went and looked, and it is across the board, the vast majority of transcripts in every assay are below 1 per cell. Right. So the vast majority of those 80% in no cell type in ENCODE exceed 1 transcript per cell. Like, that's in their own data. So we can appeal to these burst transcript kind of things. It's maybe cell specific, but there's no data to support that as being a widespread phenomenon across the 80 some odd % transcription that's been documented in the human genome. Just like we have the data on that.
Yeah, but since 2008, lots of other experiments have been showing this burst transcription.
Well, that was from 2012. Or sorry, that was 2014. That was in the supplementals, the Kellis paper. That was 2014.
Right. But what I'm saying is this burst transcription thing is something that's important to consider. But there are also other.
Well, hang on. There was a second thing there before we move on from that. Okay. There was a second thing with that, because if something is going to be biochemically relevant at very, very low concentrations, right? Like a long non-coding RNA is going to be, 1 in 1,000, 1 in a million, whatever these very low frequency transcripts that we document. If that's going to be biochemically relevant, then I think we will probably agree that it has a very specific target that it's associating with, right? It's not just binding willy-nilly with it's not bumping into stuff and sticking. It's doing a highly particular job that's affecting like a specific enzyme, a specific pathway, something like that, right?
Yeah, so there are certain transcripts where the address is the transcript. You don't want to have the transcript go far from where it's made. And so in the end, you need very low levels for that kind of...
But that is not the question I just asked. That is, to be clear, what I'm saying is in cases like that where you need very low, where very low amounts would be biochemically relevant, is what I'm saying. If very low amounts of a transcript are biochemically relevant, then that transcript is likely highly specific to its target. Because if it had many targets, then the low level would not do the job, right? Does that make sense? You're distributing a small amount widely, it's not going to do a job. So to
Right.
say.
Well, and if the transcript is the main functionality, I mean, we know, for example, that certain genes, like what is it, AIRN, in mice, the transcription of that actually stops the transcription of the down river gene,
Right.
and then that transcript's never used. It's digested very quickly. So in those cases, we have extremely low levels of transcriptions, but nevertheless, the transcription of that part of the genome is absolutely necessary.
So you're describing the thing that I've described. That would be an example of something that is active at low levels and has a very particular target, right? It's binding to that spot in the genome. It's not grabbing onto other stuff.
Well, but the transcript isn't binding to anything.
So then describe biochemically what it's doing then.
So by transcribing that part of the genome, it stops the transcription of the gene that's just downwind.
It prevents RNA polymerase binding to the promoter.
Right.
Downstream.
So, right. Exactly. So, and I give you a couple of examples of this because there are several.
No, I see. I gotcha.
But
So
okay. In those cases, the transcript's not used at all. It's digested very quickly. So there's virtually none of it there.
Gotcha. So in that case, you need that sequence to do its job. Yes. Like it has to have a promoter that says start here.
Yeah, but a lot of the downstream stuff is not necessary. So
sure, but we're talking about
of that entire section, only a few parts of it is sequence specific.
But we're talking about constrained against deletions.
Okay.
So that would fall into that, right?
As long as that part wasn't deleted. Other parts could be deleted and it should still work.
Right, but you need the region where it's transcribing actively, right? You can't get rid of that. Even if the sequence doesn't matter, you still need the region, right?
The beginning of it, at least, right?
Well,
I'm not sure that the length matters. I'm not sure about that. It could be the length matters, but I don't think it does.
Okay. Because if it's just the promoter and like a little bit downstream, then that's going to be like a minute section of the genome because those are teeny.
Well, I mean, but that's what I keep hearing. Well, yeah, this is an exception by general rule, but it's small.
Well, yeah, that's how it works, right? If we're talking about 100% of the genome, and you could say, okay, we've got all the herbs, right? And here's 10 examples that do a function. Well, what about the other 200,000?
Right. Well, because once again, we could have initially said none of the pseudogenes or none of the herbs have function. We now know some of them do.
Some of them do.
And this is why Stitz et al. Said the days of junk DNA are over because they kept finding
So what are the other things? So what are they?
Well, according to that paper anyway, they're finding that they were surprised at some of the functions they found in these mini-sats, right? So in the end,
again, those are very small amounts of the genome.
Finding these, okay, but once again, I guess why would Stitz be so convinced, and her colleagues and other folks who are evolutionists, why would they be convinced that the days of junk DNA are over if this is so non-controversial?
I think it's very controversial. I also think it's very clear is the thing. And I think my honest answer to that question, I think a lot of people got taken in by the ENCODE PR is what happened. There's a significant misunderstanding of some basic like biological concepts inherent to ENCODE, which you see in a lot of this stuff. And for example, the distinction between mutations and substitutions and the activity of purifying selection in the genome is absent from a lot of that consideration. Now, the 2014 paper actually fixed that in a lot of ways by considering constraint in their evaluation. They actually like lay out criteria of what they think. And again, this was an ENCODE paper. It was 2 years after the famous one, right? Where they say, here's what we think functional regions look like, right? It's gonna be highly conserved or constrained. They kind of go back and forth on it. I think constraint is a better word because that implies purifying selection. It doesn't, you can have regions that are constrained but not conserved across like all mammals, for example. So you have a region in the human genome that's constrained and/or has high levels of biochemical activity XYZ, right? Transcription, specific protein binding, whatever. And we're distinguishing things like specific protein binding, like a histone binding site, for example, or an RNA polymerase binding site, from the much more generalized happenstance binding that just occurs among biological molecules, which is a lot of the genome. Proteins stick to it by chance, but they don't do anything when they stick to it. They kind of attach and they bounce off and they go somewhere else. So they laid out, like, here are the conditions that we say are the criteria for function, assessing a region as functional. And they say, and here are the criteria for assessing a region as non-functional. And they say low constraint and low activity. Right? Those are just, that was in that 2014 paper, right? That's what they said in that paper. Yes? Agree?
Well, they said that these are the ones that are unambiguous.
Right. Yes. Yes. They said, like, well, I don't know if they used the word unambiguous. They were,
well, I don't think they used that
they were
word.
pretty hedgy.
But what they were trying to say was, here's where some people think our definition of function is wrong. So we're going to raise the level of that definition a little bit.
All right. Well, they didn't frame it that way, though. They said, like, here's how you would conclude that something is non-functional. Like, that's how they described it. And they said low constraint, low activity. And if you check those boxes, we think, in code, it's non-functional. Right? That's what they said in that paper. And then in their data of that paper, you look at it, you look at the transcription, you look at the protein binding, you look at the conservation numbers. Well over 2/3 of the human genome meets the non-functional bits.
Yet the folks that in code haven't come out and said that well over 2/3 of the genome is not functional. So no, I think they're interpreting that paper wrongly. They
specifically said in that paper, we're not going to give you a number, but here's the data. And then if you look at the data and say, okay, here's what they say, right?
You look at the data.
You can read. So you can say, here are the words they say, right? Here are the thresholds we're establishing. And then you look at the data and you see what parts of the genome are beyond which thresholds. And
once again, that's why I'm saying they're simply raising the definition to be a little less controversial.
So you think the definition of function is any biochemical activity then? The earlier definition?
I think transcription is a better measure of function. I mean, obviously the best measure of function is to go out and find the function. With 3.3 billion base pairs, that's a little hard to do. But nevertheless, I do think transcription is one really key ingredient that makes it more likely than not that it's functional.
And now we get back to the bioenergetic cost issue, right? Because if transcription is the standard, we can directly show that purifying selection cannot prevent spurious transcription. That is something we can directly empirically document. Do you understand that?
Go say it again.
That natural selection in humans, given our effective population size, cannot weed out spurious transcription.
Cannot weed out spurious transcription.
In other words,
you're saying that it's that the energetic cost isn't enough.
Yeah.
I guess I would say under certain conditions, yes. Probably under certain conditions, no.
Oh, right, because that's what the paper found, right? Is that below a certain level of transcription, you cannot prevent it from happening. That's what that paper found.
Okay, I guess I'm not sure what you're saying. Sorry.
Well, because the vast majority of transcription in the human genome that we've documented is below the threshold established as selectable.
Oh, in terms of the energetic costs.
Right.
Once again, under certain conditions, yes. Probably under certain conditions, no.
Well, the conditions are effective population size and level of transcription, which is, scales with the cost, right?
Right. But so
given
a small population, it may be much more important. In certain external conditions, it might be more important. I mean, we're making very broad statements about how we can measure the selective cost of something.
We know the energetic cost, though, right? We know the energetic cost of adding a nucleotide to a polymer. Mm-hmm. We know the energetic cost of replicating a DNA molecule. We know the energetic cost of doing translation, because that was the other really interesting finding in that paper, was that while most transcription, right, is below the drift barrier, so you can't select against most of the transcription that occurs, translation, on the other hand, is so costly that you can prevent most spurious translation. So junk RNA would be very common, but junk proteins relatively uncommon. Mm-hmm. And so that's a specific prediction we can make, right, based on the energetics and the effective population size and our calculation of the cost threshold, right?
And I agree with that. Translation is much more costly, yeah.
Right. And we can go out and test that. We can go and look, and we have looked. We've done this for lots of different populations, saying here's their effective population size. How does that track with their genome size? And we can see that it's in line with the predictions, right?
it's no. I guess maybe I don't know the literature well enough to say that, but no, I would disagree with that.
So, I mean, a simple example is bacteria, right? Bacteria have effective population sizes that are, I don't even know how many zeros, right? Would be in the, the E. coli effective population. I have no idea, but it's, enormous, right? I think we can agree on that, right? It's a lot. And E. coli genomes are really compact and streamlined. They have very few regions that are repeats. Like ERVs are very rare. Like ERVs, and what I mean is they don't have retroviruses in the same way we do, but like viral insertions or plasmid insertions that are dead, so to speak, are very rare in bacteria, right? Those are they're less.
I don't know that, but I'll accept
they are. They're these types of things that like the 44% retrotransposons in the human genome. You don't have that in a bacterial genome. Right, it's much more compact in terms of like gene, regulatory sequence, you got the operons, and the reason for that is that at the effective population size that bacteria have, the difference between this level of transcription and spurious transcript, like that difference is selectable. At a human population size, it's not. Selection cannot operate efficiently enough to weed out the individuals with a little bit of extra transcription. Right. So we end up with genomes that are full of just junk, if you will, that gets transcribed, and there's no cost to it that is selectable. There may be an energetic cost to doing it, but it's below the drift threshold.
But I still disagree with that final conclusion, because in the end you're saying because the cost is so low, then therefore it happens a lot. It can happen a lot for a lot of other reasons besides the cost is so low. It could be that transcription is used to actually purify the genome itself. The transcription coupled repair is a way, and we know this happens in sperm, for example. Sperm transcribe a lot of genes they don't use, and that specifically reduces the mutation rate. So all of this transcription that occurs at such low level could simply be a way of guarding against these structure-destroying mutations.
But again, we're kind of going in circles here between constraint and energetics, because your argument then relies on constraint. You need to maintain the regions in order to do their job.
You need to maintain the structure, right?
Well, but the paper that I showed was purifying selection against deletions.
All right.
Okay, which means if there's no cost to deleting it, it's not doing the job you're describing, is what I'm saying.
Now, once again, you know more biology than I do, but I would have to disagree with that.
Okay, on what grounds? Because that's how that's what selection is, right? If you had a thing that did a job that was important and you lost that thing, on average, the cells, you don't even have to look at the individuals, like the cells that experience those mutations are going to either die off or replicate slower, right? So like in a germline, those mutations are just less likely to get passed on. And the sperm example is great because, again, you can look when we're looking for purifying selection, that ropes in sperm competition because we're looking across generations, right? We're looking at big populations, so you're looking necessarily across generations. That includes sperm competition, which is a major realm for natural selection operating on the human genome. So if you had these structural regions in the DNA that had to be there to like maintain the integrity of the genome, you would see those regions experiencing purifying selection in that if you got rid of them, the sperm in which those deletions occurred, because the male germline replicates super fast, right? Tons of DNA replication, which means tons of mutations compared to other tissues in your body. That's where we're going to see the constraint. We're going to see those regions being preserved across all of human population. You could think of it as over time they're resistant to deletions because when deletions happen, those sperm don't succeed.
Right.
Right. Well, so here's the thing. I just want to point out, and that'll give you, but I just want to point out you're agreeing with every step in the pathway and then disagreeing with the conclusion.
No, I'm not agreeing with every step in the pathway. I think it's very easy to measure these things with bacteria. It's much harder to measure these things with higher organisms and people.
Do you dispute the method of purifying selection as a metric for the activity of natural selection operating on a genome? Like looking at variable substitution rates across a genome?
I do if we're looking at a sequence, yes.
Well, if we're looking, so it didn't quite answer the question, because what you're looking at a population, right? Like a population of individuals, and we can see that in parts of the genome for that population, there's little or no variation. And for other parts of the genome, there's a lot of variation. Yeah. That is, so we have variable substitution rates across those regions of the genome, right? That's just what those words mean.
Right.
Do you dispute that that is a valid way to assess the activity of natural selection?
Yeah, I do dispute that, because I do think that I agree that constrained DNA, it simply shows that it is very sequence specific. Unconstrained DNA simply shows that it's not sequence specific. But that doesn't indicate it's not functional.
But the paper, the 8.2% paper specifically was looking at deletions, not single base substitutions. So it was constrained against losing the region of DNA. It was looking at regions in the human genome that are tolerant of deletions and aren't. So it's not about the sequence. It's just, does that region have to be there or not?
Right. So I don't know that paper as far as I remember.
Okay.
But I would just wonder why people like John Maddock don't take that paper and completely review revise their idea that the genome is essentially 100% efficient. I mean, you've got all of these evolutionists, or not 100% efficient, sorry, roughly 100% functional. You've got all these. You're saying this is so obvious and not debatable, but there are lots of evolutionists who would disagree with that.
Well, I think we're conflating two things there. I'm saying the way we detect purifying selection is well understood and well accepted. The absence of junk DNA is highly controversial.
Right. But what you're saying is
those are two
it's
different
uncontested things. That only about 8% of the genome is constrained, and that the other 90% seems utterly tolerant to all sorts of deletion. What you're saying is it's obvious that the 90% is not doing anything.
So ENCODE's own numbers verify the constraint findings. Like, so just at the level of like what percentage of the genome is constrained against deletions, it's not just that one. I mean, ENCODE 2014 describes basically the same thing, right? The Kellis paper describes the same thing. So that's what I'm saying is not, I don't think anyone's really disputing those numbers. Do you agree with that or not?
I guess I have to disagree with that. Now, maybe I don't know the paper as well as you do and all that.
How should we measure it then? How should we be measuring constraints if not by looking at variable substitution rates?
Well, what I'm saying is you're talking about constraint against deletion. Yes. That's different from constraint against sequence, right?
Sure.
And I'm just saying, I'm not sure how we can be so confident that 90% of the genome has no constraint against deletion, and yet there are all these other biological experts who say that 90% is still mostly functional.
Yeah, I think that is an inconsistency in their thinking, because if you ask them, I mean, you can look at ENCODE and they say you've got a bunch of unconstrained DNA in there. I don't think, to my knowledge, the very gung-ho anti-junk DNA biology community doesn't dispute the level of constraints. I mean, it's fairly well established that about 5% of the genome is conserved across all of mammals. And there's another single-digit percentage that's lineage specific for humans. Gets you to in the neighborhood of 10%. And that's like quite well established. Now, I think it is a problem for the anti-junk DNA people to look at that number and say, yeah, but 90% of the genome is functional, right? I think that is a disconnect that they need to explain, because they're not disputing the conservation number. That's very well established, is what I'm saying. I want to just like, I'm not jumping to the next thing. I want to get us to agree on the conservation number, right?
The conservation number I have no problem with. I don't think it means what you think it means, but I think the conservation number is fine.
Okay, but then the question is, what specific functions is the genome doing that do not require the DNA to be there in order to do those functions?
Well,
because if you're saying, I just want to, let me just make the point. Sorry to jump on you there. If you're saying 90% of the genome is functional, right? And we say, okay, 10% of that genome, you can't delete it. You got to keep it. If you delete it, there's a problem. And we have various ways of verifying that, and that is a broadly accepted number. That means we've got 80% of the genome that you're saying is functional, but you can actually delete it, and that's okay. So what functions fall into that
I'm not
box?
saying you can, obviously I'm not saying you can delete all of that. You might be able to delete sections of that because the DNA is very resilient.
Right.
And so in the end, you might be able to delete one section that's actually functional and still be okay. Right. You might be able to delete 1 or 2% of that 80%, and it still be okay because the DNA is resilient. What I'm saying is, just because you see there are certain regions that can be deleted and still be okay, that doesn't mean that region isn't necessarily non-functional.
Muted myself while I was typing there, so I wasn't clicking away in everyone's ears. Yeah, so you're crossing two different ways of evaluating constraint there, right? There's the biochemical, can I delete this region? Is there a cost to that assay? And then there's the population level, what level of variation is acceptable in the population assay, right? Those are both valid ways of looking at it. And the population level assay, which is the broadly accepted number of constraints in the human genome, which again, high single digits, the thing there is that includes like the redundancy that you're talking about, like that's inherent to that number. Because if there was, say, 1 or 2% of the genome that you could tolerate on average losing to a deletion without cost, then that 8% number would be 6%, right? Natural selection would then do its thing and say, oh, we don't need all that DNA. That's hurting us.
No, it's not.
So the people who have the loss of that DNA on average do better, or the sperm cells that have that deletion on average do better. And over time, we would move towards a lower level of constraint, right? That's why the purifying selection constraint, looking at the substitution rates, is such a powerful way to look at this, because natural selection sets the threshold. And if you have that built-in redundancy, natural selection is going to eat through that, and you're going to settle at a lower number.
Yeah, but once again, this is the problem. This is why I keep saying you're not defining selection properly. What you're saying is if I can lose part of that genome, then it's utterly irrelevant and natural selection is going to eventually weed it out. I'm not saying that. I'm saying we can lose part of that genome, and as long as there's another part there, everything's going to be fine. Right. You lose both parts, there's a real problem. Right. And so in the end,
I agree.
Okay.
And if that was the case, then if that was the case, that you could do that for some percentage of the human genome, right? Then instead of showing that 8%, it would be 6%, right?
No.
Because over time, you would ultimately lose. It would be a ratchet of deletions that don't have a cost.
No, because the redundancy has a value as well. The redundancy has a value. The fact that I can lose one region of the genome and not the other, the fact that I've got two chances here is really important. I mean, the genetic code is based on that. It's based on degeneracy, right?
And we're diploid, so homozygosity solves that problem. You can select against the homozygotes that have lost both regions, but you can keep the heterozygotes in the population that have lost one region. So over time, you're going to end up with one of those regions gone.
No, because in the end, both of them make you more resilient, more likely to survive stress or to survive some change in environment. But one of those regions you can still lose and be okay.
So you're positing that these specific regions they exhibit additive variations. What you're saying?
I think so. I'm not sure if I'm not sure if I understand that terminology exactly. But what I'm saying is, we see a lot of built-in redundancy in the genetic code. It's not surprising that there'd be built-in redundancy in the genome as well.
Well, okay, but you need to have direct evidence for that redundancy, right? And what we can show is that once you get to the level of constraint that humans have, if you eat into that percentage, right, that 8% that's stuck, right, that you can't delete, there's a cost to getting rid of that stuff. Let's go back to the definition of fitness, because you mentioned that a minute ago. And also, I just want to note we've been going for like an hour and a half, and I don't want to, step on your time too much. So I just want to make sure that
I am an old man, yeah.
So I just want to, the definition of fitness, very clear, very straightforward, is a certain genome's contribution to the next generation relative to the other genomes in the population, right? That's what fitness is. If you have fitness that is greater than average, your frequency of your alleles goes up. If your fitness is lower than average, the frequency of your alleles goes down. So we have our constraint at 8 to 10%, And what that tells us is that if you experience a deletion, you eat into that percentage of the genome, you get rid of a part of it, and that number goes lower, you don't do as well as your competitors. And that can be at the individual level, it could be at sperm competition level, right? There's different levels where this can happen, right? Yes, that's just what that means. Like
it means that you do very poorly when there's this 8.6% that you say that I don't know, but let's say it is this 8.6 that is absolutely constrained. Then yeah, if you lose that, you do extremely poorly. There's this rest that if you lose that, you don't do quite as well. But as long as you don't lose other redundancies, you're going to survive.
I would love to, we don't have the time, but I would love to understand how that works mechanistically at the population genetics level, right? Because what you're describing, because we're diploid, right? It's a diploid organism.
Yeah, sure.
What you're describing is selection against homozygotes for the deletion, right? If you have one deletion, you have the other deletion. Right?
No, I'm talking about two seemingly unrelated.
You're talking about two loci in the genome. You're talking about haplotype heterozygosity. As long as you maintain the haplotype heterozygosity, you're good.
I'm not sure I understand that term. Once again, you know more biology than I do.
It has to do with linkage, right? It's if I have allele A over here, what allele do I have over here, right? It's the link. That's haplotype heterozygosity. It's a measure of equilibrium.
I'm talking about something more fundamental than that. Let's suppose.
I'm sorry to interrupt. It's just that's the thing that I'm trying to get this to where I can quantify it, because there are population genetics concepts for all of the things that you're kind of describing. But every time I try to describe one of them, you're like, no, it's not quite that. It's like, well, what specific mechanism are we talking about then?
So I can give you one example of what I'm talking about. I don't know if this is reasonable or not, but let's suppose you've got two regions of DNA between a gene, or that a gene's between, and when we wind up that gene, then one acts as a mechanical spring going one way, one acts as a mechanical spring going the other way to reduce the chance of another promoter being exposed that shouldn't be exposed. Let's lose one of those springs. Still, the chance of a promoter being exposed is pretty low because of the other spring.
Mm-hmm. And I get you. And then what I'm saying is what we're looking at the population level is we're already at the threshold where if there's a further deletion, there's a cost to that. That's directly what the variable substitution rate is telling us. Like, we're already there. That's what I'm saying.
Well, I'm not sure I understand or agree with that. Maybe it's because I don't understand it. I don't agree with it. But the idea is if you've got multiple redundancies, just like any design system, you can lose one redundancy and everything will be fine. And then later on, because of the way reproduction works, the genome will be you'll eventually get a situation where you're back to those two redundancies because of the mixing of different individuals within the population.
I think the disconnect here is there's some evolutionary dynamics at play having to do with purifying selection that we're not quite connecting on.
Okay. And that may be an impasse. I don't know.
Let me just say it this way. I think there's some evolutionary biology here that you're not familiar with.
I'm more than willing to concede that there's evolutionary biology I'm not familiar with, but I'm sure people like John Maddock are. So that's my fundamental disconnect here. If this is all so obvious, you've got these people who are very knowledgeable, possibly more knowledgeable than you, who say this is not the case.
Then where are the counter arguments to, because the arguments I'm making are not novel here, right? Mm-hmm. Like, I want to say that for everybody. Like, I'm not saying new things. I'm saying things that are in the literature. They're in a couple of the papers that I linked down below. There have not been responses to these things. And for the papers that were published in 2013, ENCODE actually did respond to them in the form of the 2014 paper, right? And like, you could say it wasn't a walk back, but like, just the plain reading of that paper is a significant walk back from the 2012.
I see, but that's not my plain reading of the paper.
And I mean, that's fine, but that's what the words say.
No, it's not what the words say. That's the thing. That's where our disconnect is.
Okay. And there have not been responses from the junk DNA is dead crowd to the kind of post-encode critique papers. There have not been a lot of times you'll see like a back and forth. Like a good example that I really love with this is there's a famous paper by a guy named Andy Kern, Kern and Hahn, I forget the year, looking at the relative importance of drift versus selection. And it was a really cool paper, and it led to like a whole bunch of back and forth of disagreement. It was really good, right? You didn't actually see a lot of that. You got the Encode 2012, you got a couple papers in 2013, the Grauer one and the other one. And then you had the Encode 2014, which basically conceded a lot of the points that were being made there in terms of incorporating more evolutionary principles in terms of evaluating functions, specifically with regard to conservation of the genome. And then they kind of moved on to other stuff. So you got like the 4D nucleome project. You've got like, they didn't keep going back and forth on that, right? So you could say like they haven't convinced all these people, but that same group hasn't really defended their original propositions. And it's those original propositions that I see coming out of like the really militant anti-junk DNA camp. And most creationists find themselves in that camp. And so when you say like, well, why hasn't it convinced a lot of people? I don't see the evidence that it hasn't, because they haven't really like set most of those, like that's a huge group, the team, most of them haven't really kept making this case in the literature in the 10 years since then, is what I'm saying.
Yeah, except that there are people that aren't in code that are still saying this.
Sure, and
right?
and yeah,
and that aren't even referencing in code when they say these things.
Right, but that's like these smart people say this thing is not an argument, right? I'm giving you very specific arguments, right?
I agree with that. But
and I'm addressing the arguments that those people are making.
Oh, but what I am saying is you're saying these are all obvious arguments that can't be argued against, but here are some people who clearly do not accept those arguments.
Okay, yeah, sure. There's lots of people don't accept things.
People who are
true.
knowledgeable, right? But you're saying this isn't controversial. It is controversial.
No. See, you're confused. That's the second or third time you've conflated the two things. What I'm saying is not controversial. The two things that I've said are not controversial is how natural selection works. Actually, there's three things: how natural selection works, how we can evaluate selection occurring in a genome, and the amount of the human genome that's subject to selection. Those things are not controversial. Now, what is controversial is how much junk DNA is present in the human genome, right? That's what's controversial. But no one disagrees on that underlying stuff. If you ask the really staunch anti-junk DNA people how much of the human genome is evolutionarily conserved or how much of it is constrained, they're going to give you, depending on how you ask the question, a number between 5 and 10%, right? And that's just what that number is. That's just a fact. And then what the job of the junk DNA doesn't exist crowd, the job that you have to do is reconcile that number with the genome is 80 or 90 or close to 100% functional. Because what you need then is a bunch of functions that exist in regions for which there is no cost if you delete them, right? That's the thing that needs to be resolved by the people arguing in favor of junk DNA. And redundancy doesn't do it because the level of purifying selection that we observe, the variable substitution rates and the regions experiencing the selection, that accounts for the redundancy.
No, it doesn't.
It absolutely does.
Yeah. So I once again, you know more biology than I do, but in the end, I don't see that in the data. I don't see that in the papers.
Okay, but I just want to point out, like, that's not an argument against the points I'm making.
Oh, I think it is, because what I'm saying is the points you're making are too hyperbolic, maybe. It's not the case that if you delete something and it has no measurable cost, that means it's not functional. That doesn't mean that.
Again, well, I mean, that is kind of literally the definition, though.
No.
Like that literally is, if it's doing a job for the cell that is necessary for that cell to operate, and you delete it, and there's no cost, is that job necessary for the cell to operate?
Doing a job for the cell and doing a job for the cell that makes it necessary to operate are two different things. Doing a job for the cell that makes it operate more efficiently or better does not necessarily mean doing a job for the cell that makes it necessary.
If it's making it operate better, how are we defining better? Because if we're defining better in terms of it's better, it's like measurably more efficient. It's measurably more competitive. It's right. If it's a quantifiable difference, then that would show up in the fitness difference and it would be selectable. That's why that's a useful standard.
No, it would only be measurable selectively if we're using all the right conditions, just like in
But we're
these.
using all of human population, though. We just keep sequencing genomes, and those are that encompasses all of humans, right? We sequence all the genomes we can, and we build this giant data set of genomes, and we look at where there's a cost to mutations and not.
No, we look to where there's high variation and where there's not.
Which
not
is
where
the
there's
same
a
thing.
cause. No, I disagree.
But you agreed earlier that's how we measure selection. And nobody really disagrees with that. Like, that's how we measure selection.
I didn't. I'm not sure. We've been round and round on this, so who knows? But no, if you said it differently before, I would definitely disagree with the idea that simply because it's variable means there's no cost.
If it's variable, that means there's no cost to having the variation. It's literally able to tolerate all those different variants.
No, I disagree with that.
Then there would be selection against the variation, though, right? That's what selection is. And it would be at lower frequency.
Depending on things like redundancy, depending on whether or not we're really testing all the different possible variables. No.
You're talking about biochemical assays, and those are, like, we're talking about two different things, right? I'm trying to get across this a population genetics thing, where you can evaluate the degree to which mutations are tolerated in a region by evaluating the level of variation in that region.
Right, but that still doesn't imply non-functionality, because lots of functional regions can tolerate a high amount of variation.
And then you can also look at how those regions tolerate deletions. Right? You could do the same thing for deletions, right?
Right, and once again, if you have redundancy, portions can tolerate deletions very well.
And so this is, I'm not going to get an answer because I've already asked this question and I didn't get an answer. But then the next question is, okay, you need to explain then the selection dynamics that enables you to preserve that, right?
Yeah, well, I'll give you an example. Let
me give you an example. Elephants have 20 copies of a gene called P53, right? That's a tumor suppressor gene. We only have 2 copies. Now, the reason elephants have 20 copies is because they're a lot bigger than most other mammals. So there's more cell divisions between zygote and adult, which means the likelihood of getting cancer earlier in life when it's going to impact your reproductive output is much higher in elephants, right, compared to humans, for example. The lifetime risk of cancer in elephants is about 1 in 30. It's about 10 times higher than humans. The lifetime cancer risk is about 1 in 3. The reason for this is that those P53 genes in elephants are redundant, right? Because in humans, if you break both copies of P53 in the same cell lineage, that's well on its way to becoming a tumor, right? In elephants, you need to break all 20 copies of it in the same lineage. So in humans, you can because most human cancers, because we're smaller than elephants, and therefore the likelihood of cancer prior to reproduction is very low in humans, right? Make sense? Okay. So there is no benefit in humans to having 4 or 6 or 20 copies of P53 in terms of your average reproductive output. That makes sense?
Sure.
Elephants have the redundancy. They have 20 copies of P53 because if you have fewer than that, there is on average a cost to your reproductive output, right? Because you're more likely to get cancer before you're an adult and therefore you have fewer kids. That makes sense?
Yeah.
Now, if you delete any single copy of P53 in any single elephant, that doesn't hurt them, right? On average, that carries no cost. They are redundant and it carries no cost. So in the biochemical assays you're describing, that's not going to pop as functional, right? But in the purifying selection assays that I'm describing, that sequence pops as functional because you can actually document that 20 versus 19 versus 18 copies of P53, there is a fitness cost to having the fewer copies. If you experience a deletion, you experience a fitness cost. So they have the redundancy, and we can detect the redundancy specifically through the test that I am describing as the criteria for evaluating function. Nobody is saying that 19 out of those 20 copies are non-functional. They're all functional. They're all doing their job. In humans, you don't have that because there's no benefit to having additional copies of P53. So if we were to have a duplication of P53 and subsequently a deletion of it, there would be no detectable fitness difference there, right? That's how this population level evaluation of purifying selection catches the things that you're describing, which is how we know that that's not what is going on in those regions of the human genome that are not within that purifying selection against deletion region, right?
No, that's not
how we know.
That's not correct. I don't think.
That's literally how we do it. It's exactly the same.
I have no doubt that there are situations where you can see the fitness cost of losing one of those genes. I have no doubt about that. But I do think there are parts of the genome that are much more resilient than that, and you don't see that. You can't see that. You can't measure that.
But so explain to me the difference, though, because if you take an elephant and you delete 2 copies of P53, there's no cost to that, right? They have 18 other copies. It keeps doing its job, no cost, right?
Mm-hmm.
But
Right.
over time, there's a fitness cost to having 19 versus 20, right?
Sure.
Okay,
I can agree with that.
It's the same.
But no, I agree that we can do that in certain cases, but I think we're talking about very specific issues here. We're talking about, for example, here we're talking about a gene, right?
So, but it's just
So we're talking
a region of
about
the
something
genome.
that acts like a gene. But
Doesn't matter.
A portion of it does. The way the genome operates is different depending on what you're doing. And so a gene works utterly different than a gene regulator, than a structural regulator. So you can't just say, okay, because this works with genes, it's going to work with other parts of the genome.
It works different isn't an answer, but that's not an answer. You need like a criterion for evaluating that difference.
They work differently, so I would expect the selection criteria to be different.
Well, the selection criteria is just based on the effective population size. Like that's what sets the threshold. Mm-hmm. So it's not about biochemically how, again, this is the point. When you're looking at selection, you're not looking at what it does. It is agnostic in terms of what the sequence does. You're able to say this region of the genome is important because there's a cost if we lose it, right? That is totally agnostic to the question of what does that region do. Do you understand that distinction?
I disagree with that.
Okay, but then explain why. Like how would, if there's a region we need that codes for a gene, if we can detect redundancy in protein coding regions through the method I just described, we agree on that.
Yeah.
Okay, what if there was a long non-coding RNA that operated through exactly the same thing? Some species, you're diploid, you get 2. Other species, for whatever reason, have 10. Right? How would that be different? And we wouldn't be able to evaluate it there versus the protein coding gene.
Well, it depends on how that long non-coding RNA works. If that long non-coding RNA is not used at all, but is instead just digested immediately because it's the act of translation that causes the regulation, then the long non-coding RNA wouldn't matter.
So you mean transcription there? But again, then it's the region of the genome that matters. Right? It's that region of the genome that matters. And if you deleted the region, you would have a cost, right?
Not if you deleted most of the region, but not all of it. So for example, if you leave that promoter going so that the transcription starts, you can lose the rest. So there's whole sections of the genome you can lose that's that's functional, that's not gonna cost you anything.
There's whole regions of the genome that's functional. But if you lose them,
not all of them.
Put it a different way. Yeah. There's no purifying selection against their loss.
I'm not sure I completely understand purified selection, because when I think of purified selection, I think about sequence. I don't think about deletions, right?
Well, it's just any change to the gene, right? Purifying selection is selection against a change, right? It's if you have a certain change, you have lower than average fitness.
Yeah, but if your change is sequence, then no, absolutely not. Not true, because large portion of the genome is not sequence specific.
Right, and when I say change to the genome, I could mean a substitution, duplication, deletion, any change. Right. If you have purifying selection in a region, you are selecting to keep that region the same. And you said a minute ago, I'm not sure I understand how purifying selection works, and I would suggest that is kind of a problem.
I didn't say I don't understand how I don't understand how you're using purifying selection.
Well, I mean, I'm describing just like the mainstream view of how purifying selection works.
Okay, but once again, you're hedging on whether it's sequence, deletion, duplication.
Let's go with deletion. We don't even need sequence specificity, right? Because there are definitely functions that don't have sequence specificity. Yes. We can document that 8% ish of the genome experiences purifying selection against deletions, right? We can document that. We can.
Yeah, sure.
Okay.
I agree.
Now
with
you're saying the answer to that is, but there's redundancy in the other 92%. So some fraction of that other 92%, a lot of it, is functional due to the redundancy that's there, right? Is that an accurate characterization?
There's redundancy, so there's no cost.
Okay. And here's what I'm trying to convey. And apparently I'm not doing it well, but here's what I'm trying to convey. And I was using the elephant example as an analogy. It doesn't matter if it's a protein coding gene, regulatory region, doesn't matter. If there's redundancy, natural selection will maintain the appropriate level of redundancy. Because if you lose that level of redundancy, there will be a cost to it in terms of your reproductive output, your fitness, your relative genetic contribution to the next generation, right? The elephant example shows that, where you can see there is extreme redundancy in elephant P53, right? That is an extreme example of genomic redundancy. And yet, there is a net cost over time if the elephant only has 19-fold redundancy versus 20-fold redundancy, or whatever I forget the exact number, 22, whatever it is, right? That's what I'm trying to convey, is that the thing that you are proposing as the explanation is a thing that we can directly detect. And what we can show by directly evaluating it is that it is not present in 92-ish% of the genome.
What you're saying we can detect it in genes. I have no problem believing that. I have a problem believing it.
So then the question is, why doesn't that same thing apply to any other region of the genome? Why are the rules different if it's RNA encoding?
The rules are different because the function's different.
But that shouldn't matter in terms of the
obvious.
fitness cost.
No, I disagree. It depends on what the function is.
Okay, tRNAs. tRNAs are highly redundant as well, right?
I don't honestly.
Yeah, the answer is yes. tRNAs, highly redundant. Human genome full of tRNAs. You've got lots of copies of all your tRNA genes. So these make little bits of RNA, forms into a little folded shape, and then you attach an amino acid to it, right? It's tRNA. There is a fitness cost to having, I don't know the exact number of each tRNA gene that you have, but let's say it's 5, right? I'm just making up numbers, chat. I don't know the exact number. Let's say it's 5, right? The reason there's 5 and not 4 is because there's a fitness cost to having 4 versus 5, right? We can like detect that at the population level. There's a cost to having deletions there. Do you accept that logic, right? That like as a valid way to look at that?
I don't know. It's getting pretty late. So, and I realize what you're trying to do here, but I'm saying that I don't agree with that necessarily. I really don't.
So I just want to point out for everybody here that we've kind of gone around in this circle several times in terms of how purifying selection operates and why looking at the level of constraint in the genome is a valid way to evaluate like purifying selection in the genome, and why that's a useful indicator of function. And I just want everyone watching this to appreciate kind of how this conversation has gone. And several times, Dr. Wile, to your credit, you've said, this is not my field. I'm not an expert in this field. And I appreciate that, because a lot of times I have conversations with people who are not experts in a specific field, and they do not acknowledge that or recognize that. And I like the tagline on your blog. If I correct me if I quote it wrong, but it's something, I forget the first part, but it was a scientist who is a Christian, not a Christian scientist.
Yes.
Which I like that, because it puts the science first, and the intellectual humility is a big part of that, right? Acknowledging this is not my field, I'm not an expert in this specific thing. I would ask that you extend that to this topic, because the concepts I'm trying to get across here in terms of like purifying selection and substitution rates and everything, this is the kind of stuff that like I just taught. We were talking before we started, I just did a 4-week summer class, intro to evolution class. This is the kind of stuff that comes up in that like intro evolutionary biology class. These are very basic, well-established dynamics. And the kind of flow here has been, okay, here's the thing, here's the thing, here's the thing. No, I don't accept that in this case. And I would ask that, given the clips we saw earlier with like the level of confidence that junk DNA is dead, combined with the level of the lack of familiarity with some like pretty basic evolutionary concepts here, maybe perhaps calls for some humility, I would suggest, when it comes to topics like this. That's what I would suggest.
Yeah, and I try to stay humble about topics like this, but like I said, I'm not the only one saying junk DNA is dead, and there are a lot of people who are experts in biology who are saying the same thing. So whereas I'm willing to concede that there are aspects of this that I don't know as well as you do, I know people who do know this as well as you do who say that junk DNA is dead. Now, once again, that's not necessarily an argument, but what I'm saying is, right, but what I'm saying is if it was so obvious, they wouldn't have this view. I do think you are cloaking yourself in a little less humility than you should as well, because you're seeing these are basic things that are controversial. Nevertheless, there are lots of there are other knowledgeable biologists who would disagree with you, at least in your conclusion.
I think you're right, and that's the problem, right? It's we agree on all this stuff. We agree on steps 1, 2, 3, 4, 5.
I doubt that they would agree with all.
I've had conversations with people; they very much do, in terms of like the level of constraints and how you detect that. That is extremely uncontroversial. That's what I'll say.
So, and I think my confidence not only comes from what I've read in the literature, but what I've read from these other evolutionists. So it's not something that I'm just coming by myself saying this. I'm coming this by reading the scientific literature and agreeing with some of what's in the scientific literature.
Right. And I'm going to make this the last thing I say, and then I'll let you say it, and then we can wrap up here, because you've been very generous with your time, and I really appreciate it. We're going for almost 2 hours. The arguments that I am conveying here, right, the arguments that have been made in the literature have not been responded to, right? You see a set of arguments against junk DNA that are very consistent over the years, really since 2012 or so, with a few exceptions. Again, the 2014 paper was different from the 2012 paper. The people promoting junk DNA have by and large not responded to the critiques that are being leveled against the notion that the human genome is 80 to 90% functional. They're just not. And you can go to papers from 2012, you can go to papers from 2008, you can find all of these things, but you look at the arguments in the aftermath of the 2012 paper, and you don't find people saying, here is the answer to how the genome is only 8% constrained against deletions, but also 90% functional, right? You don't find people reconciling those two things that people do hold in their heads. They believe both of those things at the same time, but they have not articulated a solution to that problem, right? So you're saying, well, why is it that people can make this argument despite what you're saying is well established? And my answer is, I don't know, because they haven't really tried.
Okay.
So so my point is, that tells me the arguments are quite strong.
Well, I would disagree with that.
Then they should publish them.
Well, then you should ask them. Right?
I have
publicly like this.
Like I see. I in the community, I do talk to people. Mm-hmm. So if you want to take just a couple minutes and give your final thoughts, and then we'll call this a night, because it is getting late.
Yeah, it is getting late. So, first of all, I am sorry if I offended you. I did not mean to be insulting you, but I do think your creation channel, Creation Myths, is an insult to me. So I felt like I had to push back on that. So sorry about that if that bothered you. I did want this to stay a friendly conversation. I think for the most part it did. So that's okay. I would say that a lot of the arguments you're giving are the kind of arguments we had a long time ago about pseudogenes, before we knew anything about how pseudogenes work. Well, we can't figure out a way this can be functional, therefore it can't be functional. However, what I see the trend of the data is, the more we look for function, the more we find it. So even if you disagree with ENCODE, I do think the trend is more and more functional DNA. Now, of course, I come at this based on my holistic view of science from a design standpoint. So I do think the cell is designed. I do think DNA is designed. So I think it's designed with multiple redundancies, multiple feedback loops, and so forth. So I do think if you include all of those kinds of functions that any engineer would put in a system, then in the end, you're going to find that the vast majority of the genome is functional. The only part that's non-functional is the stuff that has deteriorated over time because of living in the natural world and so forth. But just like a car can run, if 10% of its like the electrical system goes out, things like that, it can still run. The cell's not going to run if 90% of its genome is junk. It's just not going to do that, in my view, and I think in the view of a lot of others.
So I think the challenge then is to identify the functions.
Oh, I absolutely agree. And I'm not arguing that we shouldn't do that. I'm not arguing that we shouldn't do that. I'm saying that we've been doing that. It's been very successful so far. And so far, I haven't seen any indication that it's going to stop being.
Oh, it's absolutely. If you looked at the trajectory that's reported, it peaked at 80%. It's only gone down since then. It hasn't gone up.
No, but I'm talking about ignore encode. Just talk about what we're finding specific functions for. That number keeps going up. We keep looking at things that's not supposed to be functional, and it's functional. Even ignoring encode, the trend is in the creationist and the intelligent design favor.
I just want to appreciate that this is the first time I've heard someone argue in favor of junk DNA while saying ignore encode, and I want to just put a pin in that because that is a novel argument for me.
No, you don't need encode. I don't. Like I said, I don't like that Maria Stitz and her colleagues are using encode to make their
sure.
They're using the results of their research.
Yeah, and he gets like 8 or 10%. Yeah.
No, she says that days of junk DNA are over.
I know.
Not because of encode, but because of what she's doing, finding specific functions for specific parts of the genome that was supposed to be non-functional.
And so then we've said this will be the end, but this will actually be the end because this is something we can agree on. This is something we can agree on right now.
Good way to end.
For how much of the genome have we identified a specific function? The answer is it's like 8 to 10%.
Yeah, sure. Sure, I absolutely agree with that. So that's a great place to end. And like I said, I am sorry if I offended you. I did not mean to.
Thank you. I appreciate it. All right, folks, we are going to call it a night. Thank you all for hanging out while we've been talking about junk DNA. Actually, I do have a little housekeeping to do here. I'm not going to hold you for like a Q&A, folks. This has been going too long. I don't do Q&As with these things, but there were some super chats in here. Let me just pull those up real fast. This will take all of 2 minutes real quick. Prixie asks a question. What is the physical biochemical mechanism for the pre-programming? That's a really good question. It's beyond the scope for today.
Transposons, things like that.
Okay, Jay should check out Project Steve. Yeah, you want to talk about people who agree or disagree. You're familiar with Project Steve?
Yeah, absolutely. But what I was saying was something absolutely different from that. I'm not saying because these people believe it's true. It's because these people who are knowledgeable about it don't accept your arguments.
We have an argument from Na'a, which is a little bit of that, I think.
I don't know that.
I love the elephant argument. Thank you. I like the elephant argument too. Duplications counted as 1 or 2 functions, right? It would be if the duplicate then neo-functionalizes, it would be 2, but if it doesn't, then it's 1, I guess. And then we just had one more for, I missed it. Sorry you were here late. You can watch the replay. All right, folks, thanks for being here. Before you go anywhere, please remember to just hit one of the buttons down below. I don't really care which one. It's fine. Leave a comment, share, do all the stuff. It's all good. Thanks, everybody. Have a good night. I will see you next time. And as always, don't get fooled.